So, I'm George Drakakis. I'll be talking to you a little bit about predictive modeling of biological effects of ENMs. So, a couple of things introductory, and also for many of you that were at the workshop yesterday, a lot of this will overlap. Right, the problem is basically two things. One is the safety of engineered nanoparticles, and the second is the reach direction to reduce animal testing. So the smart way to go about this is to introduce non testing methods. These may include grouping approaches, which includes read across, will be discussed later, QSARs, which I'm sure you'll hear a lot about during the entire session, and expert systems. Another thing I want to point out is that there's a multi-perspective characterization about nanoparticles. What I mean is that this is in no way trying to claim that classical QSAR is easier, but there seems to be a lot of additional problems that are involved here. So the type, for example, is a significant part of modeling. You can't bunch up together fullerenes with carbon nanotubes, for example. The phys-chem characteristics are very diverse as well. Biophysical interaction, biological impact. I think we're going to hear a talk about, somebody's going to talk about the corona formation, biokinetics, you name it. So this needs to be looked at from a lot of different ways, and I will elaborate further in this talk. So as Barry mentioned, we are part of the eNanoMapper project, which has answered the call to try and do something with this. So we have essentially three main features. One is the data storage, second is the ontologies for the characterization, and the third, which is what I'm going to focus on, is the modeling, because this is what our working group is doing. So we have developed what's called Jaqpot Quattro for the fourth version of this. So it's the computational infrastructure of eNanoMapper, and it supports pre-processing of data, whether it's variable selection or descriptor generation, data mining, machine learning, and additional tasks such as domain of applicability. This will all be described in detail further down. I should say that it has been developed following the OpenTox APIs. Okay, so 4 slides after I've been talking about APIs, I'm actually giving you the definition there as well, which is good. This is what our API looks like, the Swagger documentation. So each of these is an endpoint, a REST endpoint, and under it are HTTP methods. So for anybody who's aware of these, they get, post, patch, and whatnot. These are, actually, I have an example here. So for example, under model, you can't see, I'll tell you. You can, for example, get all models available. You can post to a model, a particular model by its ID with a data set, and you can therefore derive a prediction. Essentially, this is what it looks like, but I'm going to, because it's a bit technical, I'm going to do most of the talk via the user interface we've developed. So, before I do that, a couple of things about what happens behind the scenes. One quite sort of significant part of this is to create data sets which actually can be modeled. So algorithms, anybody who develops them knows that they require nicely formatted data, whereas we're dealing with a lot of heterogeneous data. A bundle is exactly this. It's our resource for bunching together everything, which I will go into detail a bit later, but essentially it's experimental effects, images, structures, you name it. It's all in there, and we have to do something with this to get it in a nice format for our algorithms. So for this reason, we've created the Conjoiner Service, which essentially standardizes all our data sets. What this means is you can have experimental data, you can have a link to an image online, you can have a link to a PDB file, and what this will do will activate the relevant web services and automatically calculate descriptors. So this would be image descriptors, which I can show you in the next slide, or a minimal sum of the PDB descriptors using MOPAC, because of course, because this is a web tool, we couldn't deliver all of them. It would take too long, in my opinion. So this is what the image analysis application looks like in its user interface state. But of course, this you won't see unless you go to that web address. So there's a plethora of descriptors that can be calculated, and the user can just click or drag and drop an image and calculate the descriptors. Now how this works is you get an image, this is an ideal case of an image, but still, to get the point, you apply a threshold on the color scale, and therefore you can derive the areas in this, for example, region that are darker. So you derive the nanoparticles, and then you do edge detection and connected component analysis to make sure that you get all of them. And each of these, you can't tell, but they actually have assigning numbers. So then for each of these, we have all the calculated descriptors. And this happens automatically just in eNanoMapper if you have a link to an image, which is quite cool. The second thing is that we generate quantum mechanical descriptors using MOPAC services. Again, this is wrapped and it's behind the scenes, so you don't really get to deal with any of that. Now, the way we exchange data is we exchange JSON documents. Now, we've created the Jaqpot protocol of data interchange, which we call JPDI, much easier. This essentially allows to incorporate any algorithmic implementation. What this means is that somebody can have a web service somewhere else in the world, and as long as it complies with the way we transfer data, they can take data from us, do their calculations, and send it right back to us, and it will work perfectly. A second thing I will talk about later is that we can actually also include custom algorithms from any contributors, but I'll talk about it a bit later in this. So, at the moment we have wrappers for Weka, R and Python, and we have algorithms provided by all three of these. Okay, what I wanted to say about Weka and R and Python is that we have templates as well in our deliverables, which are public. So if the way that we take data and give data is actually even formatted for you, all you need to do is write your algorithm, and then you can contribute to this project if you feel like it. So some of the algorithms that are available have been listed here. Basically all major algorithms are exposed as web services, made available through both the API that I showed you and the user interface. And this is what the user interface looks like. So I don't know if you can read it from there, probably not. So we can create a data set, we can do nano QSAR modeling, nano QSAR validation, experimental design, interlaboratory testing, and read-across methods. I will go through each of these very, very briefly. So for creating a data set, basically we reach Idea Consult, which is the Ambit database, which has all the data for the eNanoMapper project. And you can select your substance owner from below. And then there's three steps. One, you can select however many substances you want from the data set. Two, select what sort of properties you want. So for example, you might only want physical chemical ones or some specific ones. And three, the third list is the descriptors. So these are the calculated ones. One is for the experimental data, the first one. And then the rest is if you want image analysis, MOPAC, and a couple of those which are under development. So that's the gerontology ones and the CDK. For training a model, it's very, very simple, three steps. One, you select a data set. You either have example data sets or your own data sets. The example data sets are data sets that have been made available from publications. So for example, we have, again, you can't read this, so we have from Walkie and co-workers and Gagowicz. I'm not sure I'm pronouncing that correctly, but maybe Professor Buzin can help out with that. But essentially we have the data sets in full and in the splitted forms that they did for training and validation. So these have been prepared and made available, which means that anybody who wants to use this particular data set or model can do it without having to do anything else except selecting it. Secondly, select a model, regression or classification, click next. Third, tune the algorithm, so the parameters, should an algorithm require parameter, could be latent variables, it could be an alpha or anything like this. You may add PMML transformations if you know how to write in the PMML language. You select the prediction feature, maybe if you want to scale, normalize, or any other transformation, domain of applicability, and you click next. That's the end of it. The model will look something like this, so depending on how generous the creator is with the information, you might get keywords or the publisher or anything like this. By clicking any of these fields, you can get the features that were used to train, if there was a variable selection, maybe the endpoint, and so on. Here you have delete, predict, and validate. So basically, okay, you can kind of understand what all of them do, but I'll go into predict and validate. Making a prediction, if you select a model, then all you need to do is have a compatible data set. Again, you click from the list that we have, and you get the list of compounds, or substances, sorry, the prediction and the domain of applicability if you click that. At the end, sorry, it isn't in the screenshot, but if you had scrolled further along this way, each of these has a button, which is the QPRF button. So this is the prediction reporting format, and it has been made available. So essentially, everything that we have in the database is already put there automatically. So if the data has available, say, INCHI codes or SMILES or CAS numbers or anything, it goes there directly. However, you can edit this online. This means that any of these fields is editable. So if you're missing the EC number, for example, you just click there, you fill it in yourself if you know it, and you can download it, because all our reports are downloadable as PDFs, and you're good to go. You have generated a QPRF report online. Now, for validation, you have the classical validation schemes, so training set split, cross-validation, and external validation. All the standard options available, whether you want to specify split ratio, folds, whether you want it randomized, stratified, you name it. And depending on the algorithm type, you'll either get the equivalent statistics and plots. So, for example, the report, which again is downloadable as PDF for validation, has the main statistics, the real against predicted in numbers and in plots as well, with a 95% confidence value and a QQ plot. So it's a relatively rich report for somebody who's doing a validation online. Now, the additional services which are being released very, very, very shortly are experimental design, interlaboratory testing, and read across. So I'll just briefly explain these. So experimental design essentially helps you choose your next experiment. What this means is that you can either start from scratch, not knowing anything about the variable you want to measure, and or you can have only a few values. So for the one that you have few values, we call it iterative experimental design. This will choose which experiment you should carry out, but it is very targeted, so you don't have to run a broad array of experiments. And the factorial design is completely from scratch. So, one at a time, iterative experimental design employs a leave one out cross-validation scheme for a PLS model. Now this is when you have at least one data point where you know something about, because then you can select from the rest of your list and say, okay, you should carry out this one, because then you fill in the gaps, and then you iterate over and over again. If you don't, unfortunately, you just use the input variables and make a PCA, and then that will suggest the experiments. So, optimization criteria, A, D, and I optimal, and maybe it's made easier for you if it's in a schema. So, you build a regression, you calculate latent variables, then you do the optimal, PLS optimal, and then here you get suggested what the next experiments you need to do. So you do them, you enrich your predictor column or your endpoint, and then you go and do it again. And this is just an iterative procedure until eventually you end up with a good result. So this is actually what it looks like. So at the top you see some statistics, because you supply a number of rounds, you supply the threshold of R squared, you supply whatever you want. And then here is your data points, and it tells you in red which ones are the next one you should measure. And then you go and you fill these in, and you run it again, and so on. It guides you towards this. Factorial design is when you do not have that column. So essentially you specify the variable names and types, whether you want them to be float or integer, continuous, whatnot, and the rows and columns. So essentially you just keep adding numbers that are equivalent for your experiment. So say for example this would be, for example, the temperature. You wouldn't make the whole grid of 0 until 1,000 if you're only interested in the range 30 to 40. So you do 30, 35, 40, and then the next experiment would be suggested to you. Right, interlaboratory testing. This is a method for assessing bias in labs. So there's two ways to do this. One of them is to do get a consensus value from the lab participants, which is the most popular one. So you get a collection of 10 labs, and somebody's bound to make a mistake. So you find out who's the outlier, and you inform them nicely, anonymously. And the other one is to have a reference material. So then you have something to go by. This essentially applies a couple of algorithms to transform the data, and then there's 8 statistical tests accompanied by plots. So actually the tests are probably the most important. So what our report looks like, apart from the 7 plots, is a massive table of statistics, and it has a table which says which thresholds each lab has crossed or not crossed. So depending on the thresholds, you get suggestions. So for example, lab 2 here needs to do some significant stuff about checking that the staff is competent and whatnot. The other 2 are just getting a warning. And this is one of the main features because it's anonymized. So these are collected by a contributor, and then the contributor can send this report without the actual real name. So not everybody needs to know who made the mistake, just that you correct it. The third service that we're making available is the read across. So there's, I'm guessing you all know what read across is, so I'm just going to say you use distant metric. We've got Euclidean, Manhattan, and the ensemble at the moment. And you use a threshold between 0 and 1. Of course, the nearest to 1 is the strictest. And essentially, you get a weighted function and you get a prediction based on this. We have something equivalent to the domain of applicability, where it's the confidence level. It's at the moment, it's the nearest neighbor confidence, but we are happy to add more methods as well. So what it looks like is this, which you can't really see, so let's keep moving. One tool I'd like to mention developed in our group in collaboration with, I think, the University of Maastricht, am I right? I think. Georgios from our group is the main author of this. So it's called RRegrs, and essentially this is a very easy to use tool, but it's very exhaustive when it comes to modeling and the parameterization. So it's easy to use, no statistical background is required. It takes into account various splits of data, and it takes a whole grid of regression parameters, for example, and then based on the best criteria of models, which is the R squared, unless the difference is very, very close, then you take the RMSE. And this is done over many folds and many rounds, and eventually it gives you the best model for your data set. So there's a standardized summary in the CSV format of text and the comparison between the models. And it is available on GitHub as well, so if anybody wants to download it, they can. Now, the final thing I want to talk to you about today is another tool that we developed recently. A undergrad student, actually, no, graduate student, sorry, Dimitra Danai Varsu. This is a online tool which allows to do gene set variation analysis and read across training and predicting. So I'll actually go to the screenshots. I think they're a little bit more informative. So if you have a gene set or a pathway or whatnot database, as long as you have an entree, gene name, or UniProt ID, either of the very, very standardized names, you may do the GSVA analysis. So for example, in this case, I'm sure you're all aware of the Walkie dataset, the Corona dataset. Okay. So basically, this is an enrichment of the pathways, the gene sets, sorry. So you can't see the most enriched ones, depending on the times that the protein, the gene name was present in the dataset, and also the spectral counts afterwards in the data analysis. So the second thing that it does is it generates a heat map if you, using that information, if you have a classification endpoint. So you may be able to see the green and red here. So this is the classification between anionic and cationic. And it's done a hierarchical clustering. Here are the enriched genes, and here are the instances. So basically, it's managed to cluster quite nicely the anionic and the cationic. And here is a cyclic graph, which, depending on the parameter you've chosen, will either show the parents or the children of the significant enriched gene sets that you've specified. The second is the read across train and predict. Now, this is very important because it takes into account different types of information. And we're coming back to the multi-perspective characterization of nanoparticles here, because you can train, but then in the testing screen, you can't see that if there's not enough information, if something is not near enough to make a prediction, it will tell you please reach in the parameters for either the biological or the physical chemical. And I'm going to come to this right now. So there's the same data set that we're talking about, the Walkie data set. It contains, I think, 50 or so physical-chemical properties and about 70 when it's reduced biological counts. So the first matrix here is all the data sets, all the variables. The second one is using all the biological, but only 12 of the physical-chemical descriptors. This is from a report by Christoph Helmer. And the bottom has been done with all the physical-chemical, but a couple of clustering algorithms that we used before. So what this does, which is the essence of the application, application is that it takes different thresholds for the physical, chemical, and different thresholds for the biological. And you have to be similar in both these areas to actually qualify to have a read-across prediction. And, okay, not all nanoparticles make it through. So you see, for example, here, only 17 of the 80, I think, made it through. But by doing this, you're refining the predictions. And if you look at the classical QSAR models, these are the best models reported in the literature, with R squared from 0.61 to 0.86. And even though we have a couple of reduced instances, you can see that the numbers that are reported here with high confidence are actually really high. So this is just something to think about, that maybe instead of bunching all information together, maybe you need to actually include lots of information from different sources but with different thresholds, because it's apples and oranges. So it's available online if you want to use it. The source code is available on GitHub, and this is the person responsible for creating it. That's the DOI for the software. And I think I'm done. I just want to thank Harry, who couldn't be here today. Harry's our invoice because of a health issue. Philip Doganis in the background. All my colleagues, Haralambos, Georgios, Angelos, Evangelia, and Dimitra that created the last app. Nanomaterials for funding, Barry and Lucian for the organization, and everybody else. And you for listening. Thank you.